Okay, let's dive straight into this explainer. Today we're unpacking Chapter 11, Reproduction and Developmental Biology. And honestly, we are embarking on what is basically the ultimate biological hero's journey, a journey that literally every single one of us has taken. We're talking about the miracle of life, tracing it all the way back to its absolute fundamentals. We're going to explore how a single-celled set of raw materials transforms, divides, and meticulously organizes itself into a massively complex, living organism. It's a process filled with incredible precision, complex chemical dances, and some truly mind-bending evolutionary strategies.
To map out this incredible journey, here's our agenda for today. We're starting with the basics of reproduction and gamete creation. Then we'll move through the menstrual cycle and fertilization, track embryonic development, and finally, look at some pretty wild animal strategies.
So, Section 1, the basics of reproduction, or getting our raw materials. Because to build our biological house, we first need to understand how the raw materials are actually sourced. How do organisms replicate their blueprints in the first place?
Well, we generally see two major avenues in nature. First up is asexual reproduction. This includes things you're probably familiar with, like binary fission in bacteria, budding in yeast, or regeneration in flatworms. But what's absolutely fascinating here is a process called parthenogenesis. This is where an entirely unfertilized egg develops into a fully viable embryo. A great example is the honeybee. Unfertilized haploid eggs, meaning they only have half a genetic blueprint, actually become males, while diploid eggs with a full genetic blueprint become females. We contrast this with human sexual reproduction, which strictly requires the fusion of two haploid gametes, a sperm and an egg, to create a brand new, genetically unique diploid offspring.
Moving on to Section 2, creating the gametes, or gathering the materials. Let's look a little closer at those haploid heroes of our journey. How does the male body, for instance, manufacture and transport these highly specialized cells?
There is actually a brilliant little biology cheat code to remember this journey, 7UP. It perfectly maps the chronological pathway sperm takes during maturation and transport. So it starts in the seminiferous tubules, where they're formed. Then they move to the epididymis to mature. Upon ejaculation, they travel through the vas deferens, into the ejaculatory duct, and finally through the urethra and out the penis. The lowercase n in 7, that's just a placeholder to help make the acronym work. And along this journey, accessory glands like the seminal vesicles and prostate add secretions to nourish the sperm and really enhance their motility.
All right, Section 3, the menstrual cycle, preparing the site. With the male raw materials accounted for, we have to look at the other half of the equation. The female body meticulously prepares a biological construction site, the uterus, each and every month.
Preparing the uterus is this beautifully orchestrated dance of four main hormones: luteinizing hormone, or LH, follicle-stimulating hormone, or FSH, estrogen, and progesterone. The way these hormone peaks and valleys perfectly align with the development of the follicle and the thickening of the uterine lining is just incredible. It's a highly intricate feedback loop, all completely designed to create the optimal environment for a potential embryo.
We can break this cycle down into 3 sequential steps to make it easier to digest. First is the follicular phase, where FSH and LH promote follicle development, which in turn releases estrogen to start thickening that uterine lining. Second is the crucial ovulation phase, where a massive spike in LH triggers the release of the egg straight into the fallopian tube. And third, the luteal phase. The empty follicle becomes the corpus luteum, pumping out progesterone to deeply enrich the uterine lining. Of course, if implantation doesn't occur, these hormone levels crash, the lining sheds, and the whole cycle starts anew.
Which brings us to Section 4, fertilization and conception, or drafting the blueprint. What happens if implantation is destined to occur? Let's get right into the inciting incident of our epic biological quest.
The absolute crucial point to understand here is that fertilization is not just some random physical collision. It is a highly specific biochemical lock-and-key mechanism called the acrosomal reaction. The sperm's actin literally has to bind perfectly to a specific protein on the egg called ZP3. This mutual recognition is what ensures that the sperm and egg are a perfect same-species match. Once that bond is confirmed, the sperm releases hydrolytic enzymes from its head to digest right through the egg's outer barrier, the zona pellucida, allowing their genetic material to finally fuse. It is just wild to think that this incredibly specific, microscopic event is the exact moment your own unique genetic code was finalized.
Now, zooming out to the geography of this whole event, there's actually a super common misconception that fertilization happens in the uterus, but this magical acrosomal reaction really happens high up in the oviduct, better known as the fallopian tube. Once the single-celled diploid zygote is formed, it begins a multi-day journey down the tube, actively dividing as it travels, before eventually seeking a place to implant in that rich lining of the uterus we talked about earlier.
Okay, Section 5, Embryonic Development. Building the house. The genetic blueprint is officially drafted. Now, the embryo has to lay its foundation through an explosive burst of rapid cellular division.
The incredibly fast pace of early division here, known as cleavage, is fascinating. What's amazing is that during cleavage, the total mass of the cells stays exactly the same. The initial giant egg cell just gets carved into smaller and smaller pieces. It becomes a solid ball of roughly 16 to 32 cells called a morula. Then, right around the 128 cell mark, it hollows out in the center to become what's called a blastula. Finally, the cells begin to wildly reorganize and literally fold inward on themselves in a stage called gastrulation, which creates 3 distinct layers.
And those 3 layers formed during gastrulation, they will go on to become every single part of our bodies. Think of this exactly like constructing a house. The outer layer, the ectoderm, forms our exterior features like skin, hair, and nails. But it also acts as the wiring by forming our central nervous system. The middle layer, the mesoderm, well, that forms our structural beams and plumbing, our skeleton, muscles, and cardiovascular system. Finally, the inner layer, the endoderm, creates the interior lining of the house, our digestive and respiratory tracts. Moving into our interior design phase, let's see how this actually builds into complex organs. During a process called neurulation, the mesoderm forms a rod called the notochord, which kind of acts like a biological formant. It literally signals the ectoderm sitting right above it to thicken, fold inward, and pinch off into a hollow neural tube. And that simple, folded tube will eventually become your entire brain and spinal cord. No way, right? It's incredible. And finally, Section 6, Animal Reproductive Strategies. We've spent a lot of time zoomed in on human cellular development. To wrap up our explainer for Chapter 11, let's zoom all the way out to see how the rest of the animal kingdom handles this incredible process of generating life. Because environments vary so wildly across the globe, life has evolved 3 completely different reproductive strategies depending on the need for protection and maternal nourishment. We've got oviparity, where offspring develop in eggs that hatch completely outside the mother's body. Then viviparity, which we humans use, where the offspring develops inside the mother and gets nourishment through a placenta. And finally, there's an amazing hybrid called ovoviviparity, where the offspring develop in eggs relying entirely on a yolk sac, but those eggs actually hatch inside the mother's body right before birth. We can see these strategies in action all around us. An oviparous chicken lays an egg into the environment, letting it develop externally. A viviparous human provides continuous internal nourishment through a placenta. But look at the ovoviviparous shark. The eggs are held internally for maximum protection. The baby sharks nourish themselves entirely from the egg's yolk, and then they emerge from the mother, appearing as a live birth. It's an absolutely spectacular evolutionary workaround.
So, what if your sex wasn't determined by chromosomes at all, but by the temperature outside? I want to leave you with this wild, provocative fact. We've talked so much about genetics and hormones dictating development, but for certain reptiles, like some sea turtles, sex isn't determined by X and Y chromosomes at all. It's literally determined by the temperature of the sand where the egg is buried. In these turtles, cold temperatures strictly produce males, and warm temperatures produce females. It just goes to show that developmental biology isn't just a rigid set of rules. It's full of endless, wonderful, environment-adapting surprises. Thanks for joining me on this explainer, and keep exploring the biology all around you.